Hi, I'm Susan Lindquist, and I'm very glad to be talking to you today. I'm a professor of biology at MIT, and I'm also at the Whitehead Institute and in the Howard Hughes Medical Institute. I'm from the Midwest, grew up in Chicago, and never in my wildest dreams did I ever think that I was going to be one day a professor of biology at MIT doing frontline research in academic translational biology. So what do I mean by translational biology? Well, I'm actually working on some problems in very basic biology that we think may help to solve some really very difficult problems in medicine. And it all has to do with those proteins that you were trying to fold using this marvelous software program, Fold-It. Fold-It gives you a beautiful picture of how difficult and complex and intricate the folds are that a protein has to undergo in order to get its shape just right so that it can function properly inside of a cell. I work on a problem of translating that kind of protein folding into the place where it's actually happening in a living cell. And that's a really kind of crazy place for proteins to be trying to fold, because it's extraordinarily crowded in there. You think of a little protein and you think about getting that protein folded, but it's not happening in isolation. It's happening in an environment where it's being bombarded constantly by other proteins. So this picture here by David Goodsell is just a wonderful reconstruction of how packed together these proteins are. What it doesn't do is really show you how incredibly kinetic and energetic this whole system is. So in a living cell, the proteins are moving around like crazy. They're folding and unfolding and moving around and bombarding each other. And this extraordinary chaos actually is what makes life possible, because it allows protein molecules from different parts of the cell to come together and to do some amazing things and give off other types of reactions and pass those on to other protein partners, etc. So that energy and movement is really important to biology, but it creates, on the other hand, some really terrible problems. Getting the protein folded in the first place when it's being bombarded by all these other proteins, keeping it folded, those two things are very, very difficult problems. And the cell devotes a tremendous amount of energy to getting the folds right and keeping the folds right. Because if a protein doesn't get the fold right, two things can happen. One is that it just won't function, and so some really very important protein functions don't get executed properly. An example of a disease that's due to a protein not folding properly and not doing its job is cystic fibrosis, a really dreadful disease, children. But that's not the only thing that can happen when proteins don't get their folds right. The other thing that can happen is that they can go crashing into other proteins and start aggregating and forming great big messes, misfolded junk in there. And that stuff is highly flexible and moves around a lot and crashes into other things and gums up the work of the cells in many, many different ways. And that causes yet another whole set of diseases that can cause, for example, Parkinson's disease or Alzheimer's disease, two really dreadful diseases that we don't have any way of stopping at present. So what my research is doing is trying to find solutions to that problem. I take these proteins that misfold in human diseases and accumulate in big aggregates and clog up the brain, and I put them into a very simple cell, a yeast cell. And the reason I do that is because yeast cells can be genetically manipulated better than any other organism we have on the planet. And one of the reasons for that is actually that brewers quite a long time ago, about 150 years ago, decided they wanted to make better beer. And so they started to figure out how to engineer this organism that produces beer, and in the process created this extraordinary subject for research. So I put these proteins, people in my laboratory really put these proteins into yeast cells, these simple cells. We start looking for things that will make those proteins behave better and not misfold and not cause disasters. And then when we find those, we collaborate with a wide variety of other people at this extraordinary campus to try to bring that work to fruition so that it can make a difference in people's lives one of these days. So for example, we're working with a chemist in the Buchwald lab to try to make compounds that will prevent these proteins from misfolding that might potentially one day be used as drugs. And we've found in using the simple manipulations we can do with yeast, we've found some compounds that are brand new compounds that people haven't worked with before. They help to correct this protein folding, and now our collaborators are making hundreds of other derivatives of those to find compounds that work even better. We're working with neurobiologists to turn human stem cells into neurons and to use the compounds that we get that rescue our simple little yeast cells, ask if they'll rescue the neurons. And my gosh, they seem to be doing that. In fact, they do it pretty darn well. And then we can use this genetic manipulations in yeast cells to figure out, well, how are those compounds doing that? What are they doing? How are they making things better? Anyway, it's a voyage of discovery that crosses over between basic science, basic biology, really wanting to understand the fundamentals of how proteins fold properly, what goes wrong when they don't, but also trying to make a difference by collaborating with a wide variety of other people in other disciplines to explore further reaches of the science in terms of neurons and the nervous system and the fundamentals of how these compounds are helping the proteins to fold.